Hello and welcome to the second part of our workshop on modeling and parameter inference. So in this part of the workshop, we will talk about model reduction and using model reduction for parameter inference. If you attended the first part of the workshop, you would already recognize some of the figures here. If not, it's fine. This workshop is intended to be independent of the material that was discussed in the first part of the workshop. But just as a recap, we looked at a software tool called Biocompiler, which can compile chemical reaction network models from specifications. So in this figure that you're seeing on your screen now, that's going from left to right on this arrow at the bottom. So you have some specifications with which you can create chemical reaction network models. But as you probably can imagine, that a chemical reaction network model would have lots and lots of parameters, which would be reaction rate parameters, for example. And to validate the model or to use the model reliably as a prediction tool, we need to validate the parameters and the parameter values from experimental evidence and experimental data. And so towards that, we need to look at the mathematical model of the CRN, which is usually an ordinary differential equation. And since for synthetic biology and in general for biological systems, our measurements are limited, we often desire this mathematical model to be of a lower dimension so that we can estimate the parameters given the limited number of measurements that we have. So towards that is the first section of this workshop, which is how to reduce chemical reaction network models, or in general mathematical models, to a lower dimension so that we can do parameter inference on that lower dimensional model. So I will talk through some of the basics of model reduction in this slides, and then move on to the coding part at the end of this lecture. And I will also talk about the basics of Bayesian inference. So that's the part which is the most interesting part here, is how you can identify the parameter values or the parameter distributions given the experimental data. So in this lecture, I will basically just go through some of the theoretical details behind these concepts of model reduction and Bayesian parameter inference. I will not go into too much detail because our goal here is to quickly jump to the software and the code and get things running with model. We will demonstrate a model reduction for any given SPML file. So for that, we will use the SPML file that was generated in part one. But if you did not attend part one of the workshop, you can just think of this as having an SPML model that you may have from any other tool of your own choice. And then we will talk about the model reduction and the parameter inference of that SPML model, both using Python tools that we will demonstrate using the Google Colab at the end. But first, let's talk about the basics behind the underlying theory that has gone into these software tools. And also feel free to post your questions on the chat or interrupt me anytime. So the first tool, as I said, is AutoReduce, which is an automated model reduction toolbox that can reduce your SPML model to a model of lower dimension. And to do that, the two steps that it takes is that it can implement conservation laws and quasi-steady-state approximations. So in this section of the talk, I will go into the basics of what these assumptions are and you might already be familiar with these because these are some of the very common assumptions that we make while modeling biological systems. So, but just before that, as I was talking earlier about how model reduction is important for parameter inference, it's even clearer with this picture here. So on the left, if you have a part and component level description of a system, and this is something that you can create using Biocompiler, the tool that we discussed in the previous part, or if you can just create this detailed model of a system by hand. And if you have this systems biology markup language SPML model for this, what you would want is to find out what are the parameters that are identifiable given the measurements that you have for that system. And so model reduction then becomes essential for analysis of the parameters in this system. Moreover, if we think about any other engineering discipline, model reduction is essential for design because we usually think of any aerospace or electrical engineering. We often work with reduced models of a simple, for example, if it's a resistor network, we have reduced models for those, or if it's a an aerospace system, we have reduced models for how the trajectories go. And we do our design, a control design, based on these reduced models, and then map these designs onto the implementation where we implement the electrical circuit, for example, is implemented with all the actual electrical components. And so that's a more detailed level. So having a connection between this phenomenological model, which is what I'm referring to as the reduced models, because the reduced models usually just model the dynamics of interest. So if we are only interested in certain part of the dynamics, we model that, and we ignore or assume that the other dynamics is not affecting, or if it's affecting, we model that as some sort of parameters in our phenomenological models. But having a mapping of the phenomenological model back to the detailed model is very common and is and has been very successful for all other engineering disciplines, and we would like to emulate that success for biological systems as well. So this process of model reduction then becomes essential for design as well. But in this talk and the workshop, we are focusing more on the analysis side, which is that if you have this detailed component level description of a system, how do we identify the parameters of that system? And so towards that, we could derive phenomenological reduced models to models things that are of our interest and things that we can actually measure. And then if we have this mapping, we get this lumped parameters in the phenomenological models that actually contain the parameters of your chemical reaction networks on the left. So AutoReduce is a tool that can help to do that. And that is what we would like to demonstrate today. It can compute reduced models. For example, if you have a CRN chemical reaction network on the left, and we apply conservation laws, parameter lumping, quasi-steady-state approximations, we can derive reduced models for this system using this toolbox automatically. So I will demonstrate the tool at the end. Obviously, that's the major part of the workshop, but it's important to also understand the theory behind what AutoReduce is doing. So let's go one by one into these model reduction methods, if you want to call them. So first is conservation laws. And you might be familiar with conservation laws because they're commonly found in biological models or chemical reaction networks. And one of the simplest examples for that is the enzymatic reaction system, which has the binding and catalysis. So an enzyme binds to substrate to form a complex, and then the complex catalyzes to form a product P, and then the enzyme is free. So we model this system with conservation laws, and this is a very common practice when we are modeling biological systems with binding and catalysis. And we can write conservation laws, for example, as E plus C equals each out. So enzyme and this complex are conserved. So that's the conserved quantity, E plus C. And similarly, S plus C plus P might equal to total substrate. So for this simple system, as you can see, it's very easy to substitute these conservation laws, derive the mathematical models that are underlying after the conservation laws, and that would give us the reduced model for this enzymatic reaction system with just the equation for the product. But what if we have similar binding and catalysis, but hundreds of times? So for example, on the left here, you may have lots of different binding reactions, which may lead to conservation laws similar to the enzymatic reaction system. And once you set those conservation laws, you are eliminating one variable per each conservation law. So you end up having a simpler model. AutoReduce has been built to do this heavy lifting, because as you can see, it's not something that's theoretically challenging. It's just substituting these algebraic relationships into the model equations. But once your models get complicated, it's important that we have an automated way to do these substitutions and algebraic manipulations. And AutoReduce can do all of that. The second and the important step in model reduction is of assumption of a quasi-steady-state approximation. So AutoReduce can automatically solve QSSA problems. As an introduction for what a quasi-steady-state approximation is, consider this arbitrary mathematical model here. So say if you have two states, X and Y, the dynamics X dot is dx over dt is 2x minus y, and y dot dy over dt is x squared minus y. And if I assume that my state X is at quasi-steady state, what I can then do is I can substitute the left-hand side to be 0, because I'm saying that X is a fast state and I only care about the dynamics of Y. So I substitute the RHS of the dynamics of X to be equal to 0. And what that lets me do is derive what the formulation for X would be if it were at steady state. So this is an assumption that we are making, and this will give us an approximate representation of this full model, and that is called the reduced model. So if I substitute X back in, I get a reduced model with just one equation down from 2. And this is basically what's going on when we assume quasi-steady-state species. Again, this seems simple enough for this two-state model, but when we have complicated chemical reaction networks and we might know what species are at quasi-steady-state, we can set those species to be quasi-steady-state using AutoReduce to derive reduced phenomenological descriptions of the system. But more importantly, AutoReduce goes one step ahead, because we might not know what species are at quasi-steady-state. So we might want to explore all possible combinations or various possible combinations of the reduced models. And so AutoReduce lets you do that and then compare different reduced models with each other so that you can make the choice of the perfect reduced model given the parameter values of the full model. But the problem that is the central problem here is of timescale separation, and that's solved by using symbolic variables and algebraic substitutions for the species that are quasi-steady-state. Okay, so timescale separation. Yeah, sorry, I should have mentioned more about that. So when I say that I am assuming X to be at quasi-steady-state, what I'm saying essentially is that X is, the dynamics of X are so fast that they reach steady state quickly. Whereas, and compared to Y, which has slower dynamics, in which I'm interested in. So whenever I'm saying that a species is at steady state, I'm essentially assuming that it just gets to its steady state quickly, and I do not care about the time scale of how quickly it gets to that steady state. And so that would be the fast time scale in the model. And the slower time scale would be the species that I'm interested in, for example, Y here. So Y would be the slower species. And so we have this time scale separation. And this concept is commonly utilized to implement quasi-steady-state approximation in biological systems, because we have processes that work at lots of orders of magnitudes of different timescales. And so it is important to figure out the timescale that we are interested in. And if we are interested in a certain order of timescale, we can reduce out the dynamics of everything that's much faster. If you have a model of a system, and if some variables are at a time scale of 10 minutes, and if there are certain other variables that have a time scale of a few seconds, and by time scale, I mean their dynamics reach their steady states in that time period. So if I'm interested in the dynamics of the species that are at the 10-minute time scale, then I might as well assume that the species that go to their steady states in a few seconds will not affect my slower dynamics as much. So there is this timescale separation, and I can take advantage of that by reducing the model by assuming the fast states to be at quasi-steady-state. So that is what I mean here by timescale separation, that you reduce out the fast dynamics and retain the slow dynamics. So AutoReduce can solve this timescale separation problem. Also, you can call it quasi-steady-state approximation problem automatically. And you can explore different parameter regimes by looking at different reduced models that may be possible for a given system. So, as I hinted to earlier, reduced models can alleviate parameter identifiability issues. So what exactly is that? So for biological systems in particular, if we have only a limited number of measurements, you can imagine that you cannot really identify all the different parameters in a detailed chemical reaction network model. So for example, on the left, if this was our chemical reaction network, it might have 100 parameters. And if you're only measuring GFP, RFP, CFP as is shown in this diagram, so you only have 3 measurements. So the parameter identifiability issue is that of the fact that with these 3 measurements, you cannot uniquely identify the values for all of the parameters in this model. And so figuring out what's the possible extent to which you can identify the parameters of the system is a central problem in parameter inference. And model reduction helps us alleviate that problem, because when we reduce this model, we are essentially lumping groups of parameters together. So that the number of parameters in the reduced model is fewer than the number of parameters in the full model. And moreover, with AutoReduce, we have this mapping available to us analytically. So we can mathematically also determine what are the parameters that change together versus what are the parameters that maybe change in the opposite way, that if one increases, the other has to decrease to maintain a constant value that that we can maybe identify with the data. So in that sense, model reduction is really, and it's very commonly used for parameter inference. But what's done in the literature commonly is you have to derive these reduced models either by your experience or by hand for very simple ones. So with this automated tool, you can AutoReduce, you can implement these common model reduction techniques, conservation laws, parameter lumping, quasi-steady-state approximations, so that then you can derive reduced models for different systems and compare them together. So with that, I will move on to the parameter inference problem. And so this will be the second section of our code workshop as well later. The first part would be about model reduction that I just talked about. So I introduced the theory behind AutoReduce. But once we have a dynamical model and we know what parameters we want to identify in that dynamical model, we need to use the experimental data to validate this model and to find out the values for these parameters. And so towards that, we have developed this black box parameter inference pipeline with Bioscrape and Emcee. So Bioscrape is a stochastic simulator that can simulate SPML models. And we have integrated Emcee, which is a Markov-Chain Monte Carlo sampler, with Bioscrape to have the features for parameter inference with Bioscrape. So there's a question: how high order lump parameters in reduced models could be related to component level parameters? Okay, that's a great question. So I will go back to this picture. So for example, and you will actually see when we demonstrate the code for this with an example, we will find out exact relationships of the lumped parameters with the component level parameters in our code as well. But as an introduction here, we have, for example, on the right, this k to the power of n, this Hill function coefficient k, right? So that's the phenomenological parameter that controls the dynamics of x1. And oftentimes what ends up happening is we just use this reduced model, and this parameter k we might try to identify. But with AutoReduce, what you can get to is, if you get to this model, reduced model, systematically, we will have an analytical relationship of k to the individual, to the binding and catalysis parameters, for example, in your chemical reaction network model. So that those are the component level models, component level parameters. And we can relate those to the phenomenological parameters in the reduced models. So, and there's another question: what about techniques for model reduction using Krylov space methods, manifolds, or old techniques like Prima? What about nonlinear dynamical systems of chemical reaction networks? Okay, so there are definitely plenty of model reduction techniques out there. The three that I talked about here is because those are the three that have been implemented here. And the reason is that we are only talking about structured model reduction. So by structured model reduction, I mean that the variables and the parameters in the detailed model keep their meaning in the reduced model as well. And that is how we are able to obtain these relationships between parameters. And also, for example, if we have x1 and x2 here in the reduced models, those would actually be something that we know in the chemical reaction network model. For example, if this was the chemical reaction network model, x1 could be GFP, x2 could be RFP, just as an example. So what I'm trying to say here is we are not introducing any transformations in model in trying to reduce these models. And that is something that is has been a common approach in other engineering disciplines and model reduction for other in other fields, and also for biology. But since we want to connect the detailed model and the phenomenological models together, we are only considering structured model reduction. And so a lot of the other methods that do that introduce linear or nonlinear transformations are not considered here, whereas conservation laws, parameter lumping, and quasi-steady-state approximations do not introduce any transformations. So your meanings of the parameters and the states are actually a subset of your full dynamics. And your other question is, what about nonlinear dynamical systems of chemical reaction networks? So we are considering nonlinear dynamical models here. So you will see that none of this is limited to linear systems. And in fact, in the example that I took, I had this nonlinear dynamical system here. And for chemical reaction networks as well, we will see that the nonlinear dynamical model for a chemical reaction network can be reduced to a phenomenological nonlinear model that can be used for parameter inference. Does that answer both of the questions? And if anyone has other questions, please feel free to post in chat. Can we identify parameters to be reduced out of the model computationally? So parameter sensitivity analysis. Yes, that is a built-in feature of AutoReduce and Bioscrape. So what you can do is you can simulate your model and also compute the sensitivity coefficients with respect to all parameters in your model. And so that way you get an idea about what parameters affect the output that you are measuring or that you care about the most. And so you can attempt to identify those parameters. And yeah, I have not gone into that here, but it's definitely an implemented feature for AutoReduce and also for Bioscrape. I will post bioarchive that we wrote earlier that uses sensitivity analysis to do better model reduction. So next question, this is only after calibrating the model, right? So by calibrating, if you mean after identifying the values for the model using the data, then yes, you can do that. But even before that, I think sensitivity analysis can help you get an idea about what parameters affect your outputs the most, even though you don't have the exact values. You can, if you have a ballpark of the values of your parameters in the model, you could run sensitivity analysis, and that will give you an idea about what parameters are affecting your output. So it's a good start, but it's obviously a cycle, as you mentioned. So once you estimate the parameters from the data, so this is what you're probably referring to as calibrating the model, then you can do the sensitivity analysis again and see and improve your parameter inference pipeline that way. Okay, so if there aren't any more questions, I will move on to the parameter inference part. And again, here I'm only introducing the basic theory behind the software tools, and we will spend most of our time running these software tools next. So with Bioscrape, we have implemented this Blackbox parameter inference pipeline that integrates Emcee, which is a Markov chain Monte Carlo sampler, to implement the Bayesian inference algorithm to identify parameter distributions. And the cool thing about this is it is more or less black box in the sense that all you need is to get your SPML model, your data in a CSV file, and you put those two things into Bioscrape and ask Bioscrape to estimate the distributions of certain parameters in your model. And it's basically these three or four lines of code can run this Bayesian inference and give you out the parameter sweeps and the distributions according to the experimental data that you put in. So we will go into that, but before that, I want to go into the basics of the Bayesian inference to make sure we are all on the same page in terms of notation, because those will be used when we run the code. So the parameter identification problem, as many of you might be aware, is to identify or train your model given some experimental data. So if there is a biological process in the center here, and you create a mathematical model for that biological process using biocompiler, as was discussed in part 1, or you reduce those dynamics out and you have a reduced model using AutoReduce. What you want to do now is to train that model according to the experimental data so that you can improve your analysis and prediction pipelines, as we were discussing for parameter sensitivity analysis. So that's the part which completes the cycle. And this is the part which is also slow, because we are identifying parameter values or parameter distributions given the experimental data using Bayesian inference, which could take a very long time to converge. But there are other methods for parameter identification. The first is nonlinear optimization, which is when you try to optimize the nonlinear dynamics of your model and find out exact parameter values that maximize or minimize the cost function. There's a question in chat. Okay, I will go back to the previous slide. I will recap this just quickly. For a biological process, we may run an experiment to get some measurements. That's the data, and we want to train the mathematical model that we have of the biological process so that we can predict and analyze the system response. That's the parameter identification problem. And there are various methods that have been used, and this is a common problem in all engineering disciplines. So nonlinear optimization is when you run an optimization algorithm to find out exact values of the parameters that maximize or minimize your cost function. The limitation for this is that it's not a general approach, and usually it works well for dynamics that are convex. For example, convex optimization methods are very well developed. But in general, for nonlinear dynamics, we may not be able to efficiently optimize the dynamics by optimization algorithms, and we may not be able to get exact parameter values. And that also connects to the identifiability problem, because there might be a manifold of values which all minimize the cost function equally, because if some parameter goes high, the other parameter goes low, and the effective lumped parameter remains the same. So we may not be able to identify exact values anyways, given the measurements that we have. Yeah, with the machine learning methods, you can identify a neural network model that might have weights that could fit the data. But the limitation here is if you want to connect that to the mechanistic details, that's an open problem, and that's not really possible when you have this neural network model and not really a chemical reaction network model, because we want to identify the different parameters of the chemical reaction network. And so that's the limitation there. And so we are left with Bayesian inference methods where we, instead of identifying the exact parameter values as in nonlinear optimization, we use our nonlinear dynamical models and we try to identify the parameter distributions. Those are called posterior parameter distributions given the experimental data. And the advantage with the Bayesian inference methods is that they are probabilistic, and it's not giving you one value of parameter, but rather a distribution of possible parameter values. And so if there are multiple parameter values that all lead to the same cost function, that can also be evident with Bayesian inference. So this is what we implemented in Bioscrape using the software Emcee. And so just quickly, I will go into the basics of Bayesian inference. This is the very basics, not the details. I will not go into how the sampler is working or how the algorithm is working. Those are topics for another session someplace else. But here we are more interested in how we have implemented Bayesian inference and how to run the tool. So the basic formula behind Bayesian inference is the Bayes rule, as most of you might be aware, that we want to find out the left-hand side, which is the probability of a parameter given the data, which is equal to the probability of data given the parameters times the probability of the parameters over the probability of the data set. So in the left-hand side, that's called the posterior probability distribution. That is given the data, what are the parameter values? On the right-hand side, in the numerator, we have, if we fix the parameter values, that is probability of data given theta, what is the probability that we see the data that we are seeing? So that is essentially saying something like, what's the difference or what's the cost function? Because we can simulate the model for the given parameter values and then compare it to the data. So that gives us an estimate of how likely are we to get the data that we are seeing given these parameter values. And the probability of theta, that's called the prior probability distribution, which is the information that you have about the parameters. So what are the probability distributions of various parameters themselves? And if you take logarithms on both sides, what you can see is the log of the posterior probability distribution is proportional to the log prior plus the log likelihood. And that is what is implemented in Bioscrape. So our goal is now to compute the log likelihood, add it to the log prior, and that is proportional to the log posterior, and that is what we're interested in. So now the problem only remains, how do we sample from this posterior distribution? And that's what will give us the parameter identification problem. So I will talk about that next. If you imagine if there is a multi-dimensional Gaussian probability density, here we know what the probability distribution is. How do we simulate this process, or how do we simulate this Gaussian probability density on a computer? That is saying as, how do I sample from this distribution? So obviously, if you use SciPy, you can just create this multi-dimensional Gaussian probability density and then create samples from it. But underlying those functions is the theory behind probabilistic sampling. And the most trivial way we can think of is I can just propose samples at random, right? I can just propose randomly any samples and see if those are accepted or not, and just keep on proposing those samples, and that's called Monte Carlo sampling. The Markov chain Monte Carlo, Emcee, does the same thing, but smartly. So if it proposes a parameter, a sample, and then depending on what the probability you get, it proposes the next sample looking at the previous probability. And so that's why it's now a Markov chain Monte Carlo sampler. So I will not go into the details of the theory or the algorithm because that will take up the rest of the time. We are, again, interested in implementing this for biological systems. So I will move to how we want to use this as a tool in our pipeline. So to summarize, the parameter inference problem can be expressed as generating some number of samples from a posterior probability density, and which is the same as identifying those parameter distributions given the experimental data. So Emcee, Markov chain Monte Carlo methods, create these random walks. They're not exactly random, but they are sampling with an algorithm that is underlying that proposes the next sample given what the previous sample was. And so by solving this sampling problem, we can solve the parameter identification problem as well. So luckily, Emcee, the Python package, has implemented this algorithm. It's one of those packages. There are multiple packages out there, but the advantage with Emcee is that it can be wrapped around easily to create this black box package that we are presenting here. Otherwise, for example, with Stan, you might need to specify the model equations more carefully and the parameters more carefully. And it's not as black box as we have here, where you just get the experimental data and the model, and you can run this parameter inference. So some notation to review here is that when the Emcee algorithm is creating these chains, it is creating these walkers. So those are the random walkers that are exploring the space. So on the left-hand side is a parameter, and on the y-axis you have parameter values. So it starts at 0.8, and then it's exploring all of the different values. And so, for example, when it goes here to -0.5, this peak, what it sees is that the cost got way worse. And so then it came back down and is again estimating at places where it's finding minimum cost. So this is a walker that's going around in space trying to minimize cost. And what we do then is run multiple walkers. So we run lots of these walkers, and if all of those are doing similar things, we say that our algorithm has converged, and what we see at the end is the posterior probability distribution, because those are the values that are most likely, because those are the values that are minimizing our cost function. So for example, on the right-hand side figure, it starts from somewhere, let's say, -0.72, and then let's say it's most of the samples at the end after 2,000 steps are, let's say, at around -0.8. So then our posterior probability distribution is something more around -0.8. So that's how Emcee algorithm gives us the parameter distributions given the experimental data, because it's minimizing the cost between the experimental data and the simulations. And on the x-axis is the number of steps. So these are the two things that we can tune while running parameter inference. Any questions? If not, I will summarize Bioscript Inference. So it can take in experimental data as a CSV file from pandas, It has built-in log prior distributions that I will talk about more in the notebook. And it integrates Python Emcee to create this black box parameter inference pipeline to run Emcee and get Bayesian inference done for the given model. Quick recap for the model training using experimental data. Yeah, so let me go back to this picture. So now that we have decided what our algorithm is for parameter inference, I can recap it again. So we have this mathematical model here. We get some experimental data for that biological process. Then for the model training, we use Bioscript Inference. And what it does is it computes the log likelihood, that's the cost. So that's this term here. And the way it computes that is it simulates, so given the parameter values, right? So we have the parameter values when we are computing likelihood. It simulates the model. So going back again here, it simulates this mathematical model to get this model simulation. And then we can compare the model simulation trajectories to the data. So we compute the norm of the difference between the model and the experiment. That's the cost function. And we want to minimize that. So the Emcee algorithm keeps on proposing new samples, new parameter values at every step. And we compute the log likelihood, and the values that minimize that cost function are preferred. And so this walker explores all of the space. And once we have run this for enough number of steps, we can sample from this to get our posterior probability distribution. And with that, I will conclude this talk, and we will move on to the interactive demonstration with the Google Colab. I have posted the link for Google Colab on Slack. If someone can post it on the chat here, that would be great. And I can take any other questions now for the theory part before now moving on to the code. Sampling parameters and cost function minimization equals parameter inference. That's exactly correct. Yes. So, yeah, right here. So generating M samples from the posterior probability distribution. That is all we need to say that we have a parameter identification using Bayesian inference given the experiment data. Because we're minimizing that cost function and then running these sample chains for any number of steps. And then if it has converged, you can just sample from it. Could this pipeline handle automated comparison of deterministic and stochastic QSSA? Automated comparison. I'm not sure what you mean there, if you could elaborate more, but if you're referring to the QSSA methods that I talked about earlier, then definitely yes, that's what we will demonstrate, that we will use QSSA to derive reduced models and then run this black box pipeline for parameter inference on it. And we can run this inference with stochastic simulations. So Bioscrape, the software that is underlying simulator, can do stochastic simulations. So we can do all of this stochastically as well. Okay, I'm gonna stop share. And I will pull up the code. So this is the Google Colab for the second part of the workshop, which is about modeling and parameter identification. And in this notebook, I will demonstrate the model reduction functionality first, and then use that reduced model to identify parameters using Bayesian inference. You can find this notebook, sorry, link on Slack or also on Zoom chat here. So you can follow along with me. You can go to File and click Save a Copy in Drive to create your own copy of this notebook. And then you can change things as well. But if you don't want to change anything, just run, then I guess you can run it with the link that we sent. So there are 3 sections for this notebook. The first is the simplest one, where we will create our SPML model. And that's the model that we will use for model reduction and parameter inference. If you attended part 1 of the workshop, you may already have seen this code to create a model using biocompiler. But if you did not attend part 1, don't worry, because that's not important for this part of the workshop. What's important is that there is an SPML model, so you can ignore that part or assume that you have your own SPML file that you want to use for model reduction. The second part would do this model reduction using AutoReduce. So it will import the SPML file that we created in section 1 and apply conservation laws and then quasi-steady-state approximations to derive reduced models for that system. And then the third section finally would be to use that reduced model and use our Blackbox parameter inference pipeline with Bioscrape and Emcee to identify the parameter, distributions given the experiment data. The example that we will run through to demonstrate these steps is a simple circuit that's an integrase circuit. So you already saw integrase models in part 1. Those were very detailed models for integrase. But here we are going to go with some simpler models because we want to identify the parameters given the measurements that we have. So if you did not attend the integrase modeling, that's also fine because this is a very simple circuit that I can explain right here. So it's just flipping of this promoter. When the integrase bind to the sites add P and add P, they flip the promoter to this activated reporter plasmid that expresses YFP. And there is another reporter, CFP, which measures the integrase that's in the circuit. So there's just one plasmid that flips because of the integrases and leads to the expression of the yellow fluorescent protein here. So a chemical reaction, just checking if it worked. Yeah. So a chemical reaction network model for this, a simple one could be that the add B site, at the add B site, 2 integrases bind and form a complex. Similarly, at the add P site, 2 integrases bind and form a complex. Then we have the flipping reaction where after the integrases have bound, they flip to form at L and at R site. So that's the reaction which flips this promoter direction to create this activated reporter plasmid A, and the integrases fall off. And then the plasmid A has YFP that is expressed. So it gives this Y, which is the output YFP. So that's the simple CRN for this model. And we can simplify it even further to match with what measurements we have. So if you go back to this figure, we are measuring the integrase with CFP, and we are measuring YFP. That's from the activated reporter plasmid. So we need a variable in the model because we are not interested in what the @P and @P sites individually are doing. What we are going to do is call R an unactivated reporter plasmid. That's this first plasmid @P and @P with @P and @P sites. And then we can similarly say, similar to the above CR and above, that 4 integrases bind to this unactivated reporter plasmid to form a complex. That complex flips to form another complex with integrases bind to it. So this one. So promoter is flipped, but the integrases are still bound to it. So that's CA, and that's an irreversible step. That's the integrase flipping. And then the integrases fall off to form this activated reporter plasmid. That's A, that's this one here. And then that A gives us the reporter Y. So we can model this using biocompiler and create a chemical reaction network. I will not go into the details of this because, as I said, this part of the workshop, we are assuming that we have this SPML model ready. So you can look at this code here. It's very simple. We just create species that we described here, and then we create the reactions, all of the individual reactions with the inputs and outputs. And that's it. We write that model to an SPML. That's all we care about because we want to look at this model and reduce this the ODEs for this model using AutoReduce. So once we have the SPML file, we can first simulate this model just to look at what the trajectories are with different initial conditions, because this will play a role when we have the experimental data as well. So to simulate this model, we are using Bioscrape. The simulator, this simulator can do stochastic and deterministic simulations, and it can import SPML files. So I'm importing the SPML that I created earlier here. And I have 25 different initial conditions because I'm running a nested loop here. So what are the initial conditions? The integrase plasmid that measures how much integrase is there is an input to our system. So how much integrase plasmid we are putting in and how much reporter plasmid we are putting in. So those are the 2 inputs. And for each of those, there are 5 different concentration values. And so I have 25 different conditions in that way. And so I can simulate that to look at how the model trajectories look like under all of these different conditions. So this next part is just plotting code that we can ignore and then look at the trajectories. So yeah, I think, William, you changed the parameters here. So
yes, they look a little different now, but then the numerics don't crash at the ends because
yeah, I'm gonna do that. Okay. Yeah, the thing is with the change parameter values, it does not demonstrate what I'm trying to demonstrate. So I'm gonna add this code here with the other parameter values. Yeah, so now what I did was I just changed the parameter values, and what we see are the 25 different initial conditions. So the columns here are the different concentrations of the reporter plasmid. So on the leftmost is the 0 nanomolar reporter, and then 0.25 nanomolar reporter. And we see that the CFP, which is measuring how much integrase is there, does not change in our model. on changing how much reporter we're putting in, which makes sense. It's independent of that. But as we change to higher reporter plasmid concentrations, we see that we get higher levels of reporter expression on the second row here. And from lighter to darker are the different concentrations for the integrase plasmid. So for the lowest, it's at 0, and then we see higher integrase plasmids leads to higher integrase reporter CFP expression. And similarly, for higher integrase plasmid, we see increasing reporter expression. So those are all 25 conditions in this figure for the circuit example that we are taking in. So we have this here just to make sure what the dynamics look like, because when we reduce the models, when we get derived reduced models, we want to make sure that our reduced models perform similarly. So let's jump into the model reduction part. So we can convert the CRNs to ODEs automatically with AutoReduce, because it can load the SPML file which has the chemical reaction network. And the notation that AutoReduce uses is that X are called the states of the model. So those are the species. F is the nonlinear dynamics of the model, and theta are the parameters. So this, theta is not the notation. You'll just see params and params values. Y are the outputs, and C is the output matrix. So Y equals CX is the model for the output of the system. So we will now import the model into AutoReduce. So that's just this one line, load SPML. And I give the SPML file name that I created. And I specify here that the outputs for this model are integrase int. And so integrase is measuring the CFP, right? And Y is the YFP reporter plasmid output. So those are the two outputs. But when I run this, I get as an output integrase system. So that's the system object for AutoReduce. And it has various parameters, as we see next. For example, integrase system.x gives us the states. So as I mentioned earlier, X represents states here. So those are the species in this system. Integrase system.f gives us the nonlinear dynamics. So those are all the, so for example, for int, that's the first species, I can do F0, and I will get the dynamics of the first species. So that's the ODE form of the CRN SPML that we imported in. This system object consists also of all the parameters and the parameter values. So when I do dot params, I can see these are the parameters in the model. So these are exactly what we created in our biocompiler model earlier. And then the values for the parameters is just params values. And finally, since we set that we have two outputs, integrase and YFPY, and our states look like this. So if we have Y equals CX, then C is the output matrix that is also automatically computed as this matrix here. So it's 1 times integrase in the first row, and then a 1 in the fifth step, which is Y. So that gives us the outputs Y equals CX. And finally, we just store these as variables because we want to refer to these variables when we do model reduction. So first step for model reduction, as we discussed, the conservation laws. In this model, let's go back to the CRN that we had. So we had this binding step of R binding to the integrase, forming a complex. So we introduced this conservation law, R plus C equals R tot here. And so by doing that, we are introducing this new parameter R tot. So here in this cell, I'm just creating this new parameter as a symbol. So that's a SymPy symbol. And then I'm appending it to the integrase system. And so now this is the important part where we set the conservation law to the system. And it's simple. You just need to specify what the conserved quantities are. So the things that are equal to 0. So R tot minus R minus C is equal to 0. And if I apply this conservation law, I can eliminate one species, one state from the model. And I specify that here. I want to eliminate R. So once I have these two lists, I can call set conservation laws and set the conserved quantities and states to eliminate to give a reduced model with conservation laws set in it. The second step will be the timescale separation, where we will do automated quasi-steady-state approximation. But before that, I want to pause for a second to see if there are any questions. All right, thank you. So let's go to the more interesting step where we apply timescale separation arguments to solve quasi-steady-state approximation-based reduced models. Now, as I discussed in the theory part, I can set certain species to be at quasi-steady state. So let's say I believe that the binding reactions are fast, and so I set the complexes that I have in the model C and CA to be at quasi-steady state. And so to do that, I just need this function solve timescale separation. And I call this function here for my system object. And I can specify the slow states for my system. So I skip C and CA. If you check, before we had .x, integrase system.x was this states, all of these states. And I ask AutoReduce solve timescale separation to give me a reduced model with just these states in it. So when I run this, it gives me the reduced model. And we can check what the reduced model looks like. So reduced model.x is integrase, activated reporter plasmid, the reporter IFP, and this is just the integrase plasmid input concentration that's in here as a species. So we have these three dynamical outputs. We can look at the dynamics of this reduced model, because this reduced model object that we have gotten here is also a system object. So it has all the same things that we talked about for the system object. That is, reduced model.x gives us the states. Reduced model.f gives us the dynamics. So f0 would give us the dynamics of the integrase, and that is just this. So this is reduced already because, as you saw earlier, it was a long equation. Where was it? Sorry. Yeah, here. So we had integrase system.f. This was this equation. And now what we have done is two steps. We have set the conservation laws, and then we have set C and CA to be at quasi-steady state. So we set the dynamics of those to be at 0 and substitute those back into the equations, and this is what we get: a simplified reduced equation for the first variable. Then the next is the interesting part, where we get the equation for the activated reporter plasmid as a Hill function, which we commonly write out just by hand and experience that it could be a Hill function if there are 4 integrases binding together. But here we systematically derived that. And you see this is a Hill function because if I just do a little bit of parameter lumping here, what I see is that if I divide by A, I see R tot times K int would become my rate parameter at the front of the Hill function. Then D plus K int by A would be the dissociation constant of that Hill function. So dA over dt is equal to this Hill function. And we have derived this from that chemical reaction network model using AutoReduce. And then finally, the reporter y dy over dt is ky times A. And that's it. That's our reduced model with these 3 variables. So I will pause again if there are questions here. So what we can do, the interesting thing is that with this automated QSSA is that you can explore lots of different possibilities of what the reduced model could be. So instead of deriving just one reduced model, which we could do when we were doing these things by hand, now since this is a computational pipeline, you can just plug in things here and see if it works. So I can just plug in, let's say I don't want A. So is it able to do it? It is. So it's giving me successful timescale separation obtained for int y and IPL. So let's see what I get now. I get just these two states. So instead of the 3-state model, I can even derive even reduced order model for this system. And I see the dynamics. I don't know what that is. This is the first time I'm trying this model anyways, but we can look into it later what that symbol means in SymPy. It's a certain symbol. And then F1 would be, yeah, I would have to look into what that symbol is. But the point is that we can change these variables. Since this is a very small CRN already, there are not many options. But if you have, say, a model with 20 different species, you can try the different combinations and derive 10 reduced models, for example, and compare which reduced model works best in different parameter conditions. I actually showed this for an example of gene expression where different reduced models are possible and give better expression, better comparison to the full model.
Ayush might have frozen, but Hatem, I can answer your question. The Hill function is the final form of the reduced model that we get here. I'll say that, as Ayush was saying, though, you could do this on different models and get non-Hill functions or more complicated Hill functions. What Ayush has done is he's used some software packages to automatically do algebra for you in some pretty cool ways. And as you probably know, algebra can result in lots of functional forms. So I wouldn't assume anything general about model reduction except to say, try it and see what you get. And this makes it much easier than, say, doing it with pen and paper, which can be quite laborious.
Yeah, that is correct. Thanks, William, for answering that question. So we go back to our original model reduction. And yeah, so when we have more complicated things here, maybe 3 or 4 different binding reactions, you can get multiple Hill functions, or maybe even not Hill functions. You can get different forms of algebra here. And it actually turns out that you don't really need to check all the exact equations that you're getting, because once you have bigger models, these things start to look ugly. So what you can just do is directly simulate this model and see how the reduced model is performing. And you can do that with some utilities that are available in AutoReduce. So you can do error and robustness metric computation. And so this is what I was talking about, maybe mentioned it in the beginning, that you can compute various possible reduced models and then compare what the error is for each reduced model compared to the full model, and also compare how robust each reduced model is compared to the full model, and then decide on which reduced model you want for your system. So there are utilities in this package that can let you do that. You can run ODE simulations. So because we have derived these ODEs, but now these are symbolic expressions, we can simulate these with the parameter values that we have set in the model with some ODE simulation utilities. And finally, there's also utilities for sensitivity analysis for this reduced model, for the full model. So you can compare the sensitivities of the reduced model and the full model. So those are all possible with AutoReduce. Question, how to make this comparison. So I guess you're asking about how to make the comparison for the error between the reduced model and the robustness between the reduced models. So I can talk a little bit about that. I have posted the bioarchives that go into the details and the mathematical theory behind all of this. But in broadly, you can think of this as, if you see here, you derive one reduced model, right? What I can do is I could have a second line of code, and I could have reduced model 2, collapse model 2, and maybe I add another, maybe I add C back in, or yeah, whatever. So if I add C back in, I'm only reducing out one species now, CA. So I get two reduced models. So one is reduced model 1, and the second is reduced model 2. Now, intuitively, what you can think is that since this reduced model has higher number of states, this will be more accurate. And so that is what it will turn out when we compute the error between the reduced model and the full model. So reduced model 2 minus the full model, take the norm or whatever, you can compute an error metric. Similarly, you can do the same thing for this first reduced model and compare to the full model, and then compare the error performance between the two reduced models. So that's the error metric comparison. The second metric that is also useful is how robust each of these reduced model is. So by that, I mean, if the parameters that are underlying in these reduced models, if they change, does reduced model 1 change more, or does reduced model 2 change more? If the reduced model 1 changes more when you change parameter values, then you can say that that's not robust, but it has lower number of species. So it's better in some sense, but worse in some other metric. So you can do those comparisons with these metrics that are inbuilt in this software. The details for those are in the papers that I posted on Slack. The sensitivity analysis is another useful tool here, which you can run. So you can run sensitivity analysis for the reduced model and for the full model, and then compare those two. So comparison at the simulation level, yes. So until now, what we are seeing here are the symbolic expressions for the models, right? We are not substituting in parameter values, but you can substitute those and simulate these ODEs by solving these ODEs using ODEInt, for example. And this is what I'm doing here. So we can compare at the simulation level, how do the reduced models compare in simulation to the full model. And so I simulated it, and I see very similar kind of trajectories. With the behavior is similar, although obviously since we have reduced the model, we get different trajectories from the full model. So there is some error in it, and we can work towards what's the best reduced model that minimizes that error. And finally, to finish up with AutoReduce, we want to get this ODE out of AutoReduce because it's now in symbolic form, although it has simulations and everything in it, but we want to get it out as an SBML. So you can simply use Biocompiler or any other software to create a simple chemical reaction network with 3 steps. So one is the formation of integrase with the general propensity, that's the rate of that. So reduced model.f0, that was the integrase. Similarly, A is reduced model.f1, and then Y is reduced model.f2. And you can write that to a reduced SBML. So now we are out of AutoReduce. We started with an integrase SBML with a CRN. We are getting out with a CRN with a reduced model that's an ODE with fewer number of states. So before I jump into the parameter inference, I can take a couple of questions before I reset the parameters. Okay. If there aren't any, there is a question. What analysis can we do at this level? So at this level, you mean, I guess, at the reduced level, you can simulate the model. And for example, an example of an analysis was this one, where I derived this reduced model automatically, and now I see d plus k over a is the dissociation constant. And so that's my lump parameter. And so I can simulate the reduced model with different values for these parameters and get an idea about parameter identifiability with this reduced model. So that's one example of an analysis that you can do at the reduced model. And it's actually very helpful before jumping into parameter inference to do those kinds of analysis, to figure out what parameters you can identify from the given data. Similarly, another analysis example is the sensitivity analysis, which you can run to figure out what parameters change the reduced model most, so that you get an idea about what parameters you want to identify when you run parameter inference. And a third kind of thing that you can do before going into parameter inference is to run the simulations for the reduced model under different initial conditions, like I did right now. But maybe you want to do control design, or you want to see how this reduced model responds to certain inputs. So you can run all of those simulations and figure out if it matches your full model description or not. Another question, would it be acceptable if I can use alternate methods that keep information about the variables, components of the chemical reaction network? I'm trying to see if non-structural methods can still be comparable to structural methods. Yeah, that's a great idea, and I would be interested in implementing something like that. And I have been thinking about non-structural methods and how those can be implemented within this pipeline. Right now, there is only the QSSA and conservation laws that have been implemented, but I am definitely interested in expanding this library of model reduction techniques that can be used with AutoReduce. So it is not possible right now, but it should be possible to extend this to do that. And I am definitely interested in that for my own research as well. So we can talk about it if more later, if you're interested in working on that with me. Okay, so let's go to parameter inference now. So parameter inference is Blackbox. We have implemented this pipeline as a Blackbox parameter inference with Bioscript and Emcee. So I'll jump right into it. I talked about the theory behind Bayesian inference and what we are doing here. So the first step is to get the data. So I have uploaded this data file on GitHub, which I can get by cloning the repository, which gives me the data file. So I can import the data file using the pandas package here. And this is what it looks like. And this is the usual syntax that is also required by Bioscript Inference when we're doing parameter inference, that there is a column called time. So that's the time points for different measurements. And there are two measurements, integrase and YFP. And I have lots of different conditions here in this data frame. And this is an experiment that was done at Caltech 4 years ago, maybe, for the circuit that I described and the model that we have been looking at. So now we will use that experimental data to identify the parameters of this reduced model that we just derived. But before that, let's plot the experimental data that we just loaded in. So for the same initial conditions that we had earlier, I am simulating, not simulating, this is the experimental data. So just with the initial conditions, I am plotting the data here. So we see that similar behavior, the integrase reporter CFP is supposed to be constant, as we saw as the reporter plasmid concentration changed from left to right, but we can see that it is increasing a little bit. So that's probably an effect of retroactivity that's going on, because when you have higher reporter plasmid, you're seeing more, you're seeing higher CFP at the end here. And the YFP is showing also similar behavior as we had in the model, but now the challenge is to use all of these trajectories to identify the parameter distributions for our reduced model. The first step that I'm going to do here is to truncate the data, because this leveling off of integrase CFP is something that we have not modeled in our model earlier. So I'm going to truncate it to 160 minutes to only have the dynamics of this exponential phase here. So I'm going to do that with just the simple trick with pandas data frame, and then I can plot the truncated data frame, truncated experimental data again. So now this looks like pretty much something that we should be able to identify or fit to the model that we had earlier. So to run Bioscript Inference, we need to load the SPML model first. And that's the first step after loading in the experimental data. So we load the SPML model by just writing this model constructor for Bioscrape and importing this SPML file. Then next is the prior information about the parameters. So this is what I talked about in my presentation earlier. That we need to encode the information that we have about the parameters into as prior distributions. And this could help our parameter inference algorithm, the pipeline, because if you are giving smart priors with parameters restricted to various regimes, then the algorithm can explore that space more efficiently and quickly. But if you don't have much information about the parameters, you can also give flat priors. You can just give minimum and maximum value for the parameter, and that's called the uniform distribution. The syntax for the prior is given here that you can read. I can just take an example. So for example, if it's a uniform prior, you can have, for this parameter K1, for example, you would write in a string that is a uniform prior and the minimum value and the maximum value. That will give you a uniform distribution like that. And similarly, there are, I guess, 5 more prior probability distributions that are built into Bioscrape that you can use for your priors when running parameter inference. You can also define custom priors. So this is your own custom function that you can define as a prior, and we will see an application of this kind of prior at the end as well. So to run the parameter inference, as I mentioned earlier, it's very simple. It's just 3 lines of code here. First is I define the priors for the parameters. So I have, to demonstrate the basic functionality, I'm just showing one parameter that I want to identify. The other parameters then will be fixed to certain value. But for this one parameter Ki, I am giving a uniform prior with a minimum value of 0, maximum value of 1E4. And I'm saying that this has to remain positive. So it should ignore all the negative values that it might sample. The number of steps is the number of steps I want to run the chains for. So I'm setting that at 500. So when you do run complicated models and with a higher dimensional problem, you would have to run longer steps to assess if you converged or not. And then finally, the line that calls all the inference pipeline is PyInference. You need to tell this function what the model is, what the experimental data is, and this is what I have created earlier. my measurements, I have two outputs. So it will look at both outputs from the experiment and from the model, compare the two outputs to each other, and compute the norm of the error between the experimental data and the model simulation for the two outputs, and add those together. And the time column is just specifying what column in my data frame is time. The number of walkers is, as I demonstrated in my slide, the number of random walkers that will start. So if I have it at 5, you will see that 5 walkers started and are exploring the space. Params to estimate is the parameters that I want to estimate. So that's just this one parameter. And initial conditions is something that I've created earlier. Is because I have 25 initial conditions, this is a list of 25 different conditions. So what this will do then is it will run the same thing for all of the different conditions, and it will try to compute that cost for all conditions and minimize it. So it's trying to explore the space and minimize the cost for 2 outputs, 25 initial conditions, but right now just for one parameter, and I'm going to run this now. So you'll see that it has a progress bar and it runs pretty quickly, but you can imagine that the number of simulations that it's doing is like 5 walkers are going for 500 steps, and each step is simulating 25 different conditions for 2 outputs and computing the norm and doing all of the computation to give out the exploration results. So what happened here? It started at some value here, 0.65. Then quickly, this is called the burn-in period. It burnt into these values where it's getting minimum cost function, and then it stayed there for the rest of the time. So we can be fairly certain that if this is the only one parameter that we want to identify, given all of this data, this would be the value, around this value would be the distribution. So that's the distribution of this parameter. So that's one parameter Bayesian inference completed with this one step. So that's the basic demonstration for the parameter inference. And I will again pause here for a few seconds, because after this, we will just expand on it and we will connect this to the data and see what we can identify versus what things we are missing in our reduced model. If there aren't any questions, I will just go to the next cell, which shows the cost progress. So this is the cost function that I have been talking about, that it's trying to minimize this cost at every step here. So it started at some cost of -2600 and then quickly went to this minimum value and stayed there. And at every step, it was proposing new values for this one parameter. We can now expand this. Before expanding, there's another thing to try is if I started at a different value, I can do init seed. So this is to specify where I want to start my walkers from. So I start at a value of 100. So this is an important trick to make sure if you have converged or not. So if you started from 2 different values but converge at the same point, that probably means that you're closer to convergence. You can run this for different initial seeds to be even sure that you're converging. So like here, I started at 100 and went to similar value, as you can see. But this is just one parameter. And the fits won't be great if we just fitted this one parameter, because the output of the model is dependent on a lot of different parameters. So let's try 2 parameters now. And I will run that, and you can see it again runs quickly, about 30 seconds for these 2 parameters, and I will get similar results. So the first parameter, as we were seeing earlier, started from 100 and burnt into this smaller value. The second parameter kint is exploring like this here, and we get the distributions and the correlations of these two parameters in this corner plot. So this is to demonstrate how you can identify different parameters. You do not need to identify all parameters. If you're certain about the values for any parameter, you can just fix it and only identify a subset of the parameters, and that is what we were doing. But to get good fits with the experimental data, usually, and especially since we already reduced the model, we only have like 5 parameters. And so if we can try to identify all of those, but parameter inference, Bayesian inference takes a long time to converge. As you can see, we just had these 2 parameters and 500 steps we were getting. It took like 15 seconds, but when we are identifying 5 parameters now, so the dimension of the problem is more than 2-fold, we would need a much longer run, and that's what we have here. So we did this overnight, I think, and you can also try it on your own with this Google Colab and with some other parameters that go into ODE int to improve the speed of the simulations, and with 50 blockers. So all of the parameters are increased now because we are identifying 5 different parameters. So you can run this. It takes a long time. I think it took me 1 hour, 33 minutes on Colab, which is slower. So on my local, it would take like 40, 50 minutes. But usually it's a parameter inference is as people call it, it's more of an art, because you need to run for different priors for different parameters, and convergence is not obvious because you just get to see these plots of how they progress. You never know if you ran this for 50,000 steps, maybe this would explore to certain other regions. You can never know until you run it for 50,000 steps, right? So it's always tricky to assess convergence. So it's always also better to run for longer times. Cost norm, it's norm 2, and it's also, you can also set it, I think, norm order. So let's do that. I think it's norm order. So the default is 2, but I don't think it was called norm order anyways. Maybe order norm, I don't remember. But there is a parameter, okay, it was order norm, I think, that you can set here that will change the norm computation of the cost function. And so with this longer run, I'm going to show the result that we got with the parameter fits versus the experimental data. So as you can see, the straight lines here are the models. In our model, we did not, as we discussed earlier, our model does not change for the CFP integrase plasmid. It does not change with changing reporter plasmid concentrations, because why would it? It's not dependent on it in our model. But in reality, as you can see, it is increasing due to retroactivity effects that we have not modeled in this reduced system. And so this points directly towards the fact that we need to use, we are missing out details in our model because we are not able to capture this dynamics in the higher reporter plasmid concentrations. Although if you look at 0.25 nanomolar, it's quite close. The model simulations are quite close. Like here, all trajectories are quite similar to the experimental trajectory. Similarly for the reporter plasmid. And so there is the code here that you can run to simulate the model alongside the experimental data and get this plot. We have plotted this already for this longer run to show this. But that is it on the code side. You can try lots of different things here. With respect to reduced models, we tried some things. We can explore more with what kinds of reduced models are possible. for this example or for any other example that you may have. And you can do parameter sweeps like we were doing here and get these correlations for different sets of parameters and figure out how different parameters are related to each other, because as we discussed, parameters are getting lumped. So if some parameter increases, the other is decreasing together. So you can explore the correlations by doing these parameter sweeps using the Emcee algorithm. And you can assess the convergence of the Bayesian inference using various methods that I have written down here. And finally, a trick is to run this parameter inference for once, and then use the posteriors that you get as priors for your next inference run. So in that case, you would need to use the custom prior that I described earlier. And if you have any questions, you can always reach out to me or anyone else on our Slack channel as well. And 3 minutes to the nominal end of the session, and we are at the end of the session, so I can stop for questions.
So I was just asked a question on Slack, which I'll respond to. It's and maybe you'll have some thoughts here. Is what analysis can we do after parameter inference? And it's a very good question, and one that I'm not sure we have good answers for. I think once you get these distributions, you can try to improve your model. You can try to infer more parameters. You can also use it to help figure out what data you want to gather next. So if you're not inferring certain parameters very well, that might be a hint to the next experiments you should run. Ayush, do you have anything else you might say?
Yeah, I mean, I think, oh, can we do after parameter inference? Yeah, I was confused for a second because you asked the same question, what can we do before? Yeah, this is a good question. And I think William already summarized most of the things, but I think also that once you identify your parameters after your parameter inference run, you can, so that's essentially like you have an idea about certain module, and you can, what you can do is if that module is working in a bigger circuit, in a bigger system, you could fix these parameters that you have identified to predict what would happen if this module were in some other system. So it's like validating certain part of your system, and then it helps you improve your design. So it fits right into the design build test cycle, where once, so this where we are now at the validation part, test, learn part. So we learn the parameters and then we propose newer designs or improve the design. Yeah, tune circuit behavior to the desired one. So you, like as an example that we discussed, after parameter inference, we saw that the reduced model that we used was not able to capture the retroactivity effects. So that's also kind of analysis that you can do mathematically as well, that what kind of model would capture that effect. So that's essentially tuning your circuit behavior to the desired one, and by improving the model from the results of the parameter inference.
I'd like to make one more last comment. We're in a room full of a lot of developers and people who I think can contribute this. Bioscrape, like biocompiler, is an open source, fairly object-oriented program. So there's a lot of ways to extend the software to make it better or fit your application. For example, we have discussed, could we do parameter inference on flow cytometry data? And we have sort of schematics of how that might work, but we haven't implemented everything in large part because we don't have the data at this point. And so there's lots of ways, if you're interested in contributing to these projects, to get involved if you're so inclined.
Okay, I think if there aren't any more questions, we can end now and continue the conversation on Slack as well. We have the IWBDA Slack channel, and also we sent the Slack channel link for our software tools, so you can join both, and we are happy to discuss further.